Hi, my name is Destiny, and today we're going to look at how forces interact to stabilize double-stranded DNA. So let's first take a look at the well-known DNA model founded by Watson and Crick. In the early 1950s, biologist James Watson and physicist Francis Crick came up with their model of the DNA double helix. The structure of DNA is a double-stranded antiparallel right-handed helix. The sugar phosphate backbones of the DNA strands make up the outside of the helix, while the nitrogenous bases are found on the inside and form hydrogen-bonded pairs that hold the DNA strands together. Double-stranded DNA is an antiparallel molecule, meaning that it is composed of two strands that run alongside each other but point in opposite directions. The twisting of the DNA double helix and the geometry of the bases create a wider gap, called the major groove, and a narrower gap, called the minor groove, that run along the length of the molecule. Base pairs aren't made up of just any combination of bases. Instead, if there is an A found on one strand, it must be paired with a T on the other, and vice versa. Similarly, a G found on one strand must always have a C for a partner on the opposite strand. These A and T and GC associations are known as complementary base pairs. A large purine, A or G, is always paired with a small pyrimidine, T or C, based on the explanation of Chargaff's rules. The 5-carbon sugar in DNA is called deoxyribose. The equivalent carbon of deoxyribose has a hydrogen instead of a carboxyl group as shown in RNA. The carbon atoms of a nucleotide sugar molecules are numbered 1', 2', 3', 4', and 5'. In a nucleotide, the sugar occupies a central position and with the base attached to its 1' carbon and the phosphate group or groups attached to its 5' carbon. DNA has an overall negative charge. Due to the negative charge of the phosphate backbone and heat, DNA wants to destabilize. This breaking of hydrogen bonds is called a process. This breaking of hydrogen bonds is a process called denaturation. Counteractive forces hold the two strands of DNA together. The suggestion of neutralization is thought to neutralize the charge of the phosphate backbone and give DNA a stronger structure to hold itself together. Using a salt buffer to complete this experiment, bridges are formed between ammonium ions and DNA phosphates, which maximize the neutralization of phosphate charges in the minor groove. Localization of ammonium ions at the center or floor of the major groove shifts the overall neutralization towards the major groove. This lowers the overall pH and raises the possibility that enhanced base stacking may make the DNA more rigid and give it a stronger foundation. And these are my references.